And with that, maybe I'll start asking a few questions. I will be mostly going by upwards and chronological order, but Dr. Poyet and Dr. Iralo, if you see like a question that I might be overlooking that you really want to answer, also go ahead and pick those out. And maybe start with a question for Dr. Poyet. And that question was by Noura, who said there was a very interesting work, and they were wondering if the IgA enrichment could be caused mainly by the differences in the diet, or also whether those could be confounded maybe by some environmental factors that are specific to industrialized communities, for instance, air pollution.
Sorry, I missed the beginning of the question, but we're talking about IgA, right? And what's triggering the differences?
Whether the enrichment IgE could, like, what are the main driving factors of the IgA differences? Might it be diet, or could it also be pollution, for instance, industrialized?
Sure, I think it can be very multifactorial, for sure. But our guess, but we obviously, that's just, ideas, hypothesis, and we have ongoing experiments and still a lot of work to do, but our idea is that the antigenic profiles actually contribute a lot to these differences in IgA levels between individuals and between lifestyles. But it's definitely multifactorial, and yeah, why not? Yeah, diet, sure. Pollution, yeah, sure.
Okay, sounds good. Then we also have a question for Dr. Iralo. That one is by Sean, and he's asking, is the human use of antimicrobials correlated with the frequency of AMR genes across continents, or do you think this could also be driven by agricultural or industrial usage of antimicrobials?
Well, this is a very, very important question, and it's quite difficult to answer with the actual data, because the data from prescription and or antibiotic use in many countries, particularly in low and middle income countries, is scarce, and some countries gather that information, some others not. So at the country level, it should be difficult to perform in-depth comparisons. But we know, for example, that in Latin America in particular, that is basically food production continent, and where livestock industries are very prevalent. During many decades, antibiotic use was totally uncontrolled in these industries, and antibiotics were used during many decades as growing factors. So they were administered in the diet of animals, and of course this contributed to the selection and evolution of different AMR mechanisms that probably aren't today present in our guts. But it's an excellent question, and I don't have the answer yet, but probably we will be focusing on trying to answer this with actual data in the next few years.
Yeah, thanks so much. So there was one question by Ken. It didn't indicate the speaker, but I think it might actually be relevant to both of you. So both of you can feel free to answer that. And actually, the question was echoed a few times by other participants, and it was basically, are there any studies on differences of infant microbiomes in different underrepresented communities? While there may be differences in the frequency of breastfeeding, there may also be significant differences in the microbiome transferred from the mother.
Please, Gregorio, feel free to jump if you want.
Okay. Indeed, this question, I was just reading it in chat, because within our data set, we have a cohort of in particular, these samples are only from Mexico, but from Mexico, we have a cohort of people from the DF, from the capital. These are urbanized people, and a cohort from an isolated native community called the Mepas, that they live in semi-isolation. And within this community, we have samples from adults and from children, and we have the data from the co-housing of these people. So we know which families live together. And this is something that we didn't analyze. We didn't have the time to analyze Mexico as a whole and perform these comparisons, trying to look if these children in this community that are mainly feed through breastfeeding exclusively, and they are not exposed to other to medicines, for example, or to antibiotics, have differences in their gut. We did some preliminary analysis in comparison with other cohorts of children across the world, and including other rural or native communities. And in particular, we found some interesting patterns with the Bifidobacterium species that are those that start to colonize the gut. And in Mexico, the Bifidobacteria species that start to colonize are different from other industrialized and also non-industrialized communities in different parts of the world. So there are some species that are very prevalent that are not prevalent in other parts of the world, and vice versa. So it's something we have there, but we didn't have time yet to explore more in depth. Yeah.
And we don't have infant in our own cohort, so yeah, we will not. But we confirm that yeah, in the most isolated and rural areas, yeah, most of the children has breastfed for many years actually, and yeah, there is a lot of co-founder effects, obviously, but that's definitely have impact on the gut microbiome.
Perfect, thank you. Yeah, then we have another question by Sean. That one is directed at Dr. Poyet. And he's saying really interesting results. Any idea about the ecological pressures that enrich for coprostanol converters in non-industrialized populations? I might have expected the reverse due to what I'd expect to be an enrichment in cholesterol in the industrialized diet.
Yeah, that's a very good question. So first, the enrichment in cholesterol in industrialized diets. Our data, so we have dietary information from all the participants in our studies, and using this data, we don't really confirm a huge difference in cholesterol enrichment in industrialized diets. Actually, a lot of very rural and isolated population consume a lot of meat or eggs, for example, several times per day. So that's actually a pretty cholesterol-rich diet. So that's one thing. About the ecological pressures, so that's only hypothesis because we didn't test for that yet. But one hypothesis that we have is that so the EcoPro strain that we isolated and tested in vitro, we actually realized that it's extremely sensitive to oxygen. So one explanation could be that, yeah, the gut of the most industrialized population may be more exposed to a higher level of oxygen. If it's more leaky, or it's a little perturbed or whatever. So that's the hypothesis that we have, but that's just ideas. We need to test for that.
Perfect, thank you. We have another question for Dr. Iralo by an anonymous attendee, and they asked, do you plan to look at parasites and fungi at some point? Has any progress been done in culturing any of the newly discovered bacterial species from Latin America?
Cool. Indeed, regarding the eukaryotes, we did it. We did attempt to assembly and characterize eukaryotes from our data, but we didn't found much things. except for some blastocystis genomes that we were able to assemble and characterize. And we suspect this is because biases in sample processing and mainly DNA purification, that some eukaryotes are more difficult to, DNA from some eukaryotes are more difficult to extract. And also the relationship between bacterial and viral DNA and eukaryotic DNA in terms of the absolute quantity may bias the thing towards the bacteria and viruses. So we couldn't achieve much results on this, but it's something that is very interesting because there are very, very few analyses of the microbial eukaryotes of the human gut, and probably they are playing a very important role that we don't know. And regarding the culturing, yes, I didn't present this, but we are also generating a catalog of and a biobank of bacteria cultured from at least some of the samples. We couldn't have access to culture from all these countries, but indeed we are doing this now, and we already have a collection of over probably 2,000 strains from Uruguayan population, and we are doing more. But now we are indeed very interested in focusing on culturing Oscillospiraceae species and have a look at some phenotypic characteristics of these bacteria that are very prevalent in Latin America.
Perfect, thanks. Yeah, I'll give you a short chance in case you saw a question that you really want to answer. But no need. I didn't want to put you on the spotlight here. I'm happy to select questions. It's just I just want to give you the explicit chance in case I'm skipping something. But yeah, while you look through that, also there was one question to both speakers. There was, what do you think is the relationship between the virome and lifestyle geographic location? Is it about viruses that infect and modulate the microbiota, or does it also include host viruses changing along with the microbiota and diet?
Yeah, really not a specialist here. I don't know if I want to make up something, but
no, well, what we found about the virome was surprising for us, because we expected to identify novel viruses, but the number of new potential phage species that we uncovered was like tremendous, and they vary according to geographic location, but in particular we don't know why. There are some populations that we analyze that are very, very high in novel viruses. For example, population from Bolivia, we don't know why, is extremely rich in terms of new phage genomes among our data sets. And well, we don't have also metadata to hypothesize why this can be happening, but it's a result that is there. There are some populations that are much richer in viruses, and the virome seems to be much more volatile across populations and across individuals. And this is why probably viruses have the potential of being very powerful biomarkers for certain regions or certain populations. And about the mechanisms of how viruses can modulate, there are different mechanisms. Also, the viruses can modulate our immune systems directly. So they can act over the bacteria, controlling certain bacterial taxa, but also directly have an effect on some immune pathways. So it's very vast. Yeah.
Perfect. Maybe I'll misuse my position as a chair a little bit here and maybe butt in with a question of my own as well. So hearing both of your talks, I was also wondering about the environmental factors as well, such as many participants. So when obviously the microenvironment in the gut itself is probably responsible for a lot of differences that we observe, but then there's also like, environmental factors that affect the host itself, which again can propagate to the gut microbiome maybe. Do you think it would be necessary to maybe also start sampling the environment itself, for instance, sampling particularly foods or dietary components from different regions, or taking like air samples or any other samples from the environment where those communities live to kind of get maybe added? Or do you think that, questionnaires usually capture the majority of that variability?
Well, Mathilde, do you want to?
Sure. I mean, I don't.
Okay.
Yeah.
No, sorry. No, go ahead if you.
Sounds good. So yes, to answer the end of the question. So yeah, no, I think even with tons of questionnaires, we're missing a lot of information. So yeah, I will really love to see more environmental samples. So I'm aware of quite some groups that started to check the microbiome of dietary incomes, and particularly fermented food, for example, source of water. That's also very important. And yeah, I think that's an important next step. I think I'm missing the point of the question, but
No,
was that it? Yeah, good.
Yeah.
Well, indeed, we have a project in which since some years ago we are doing metagenomics on the urban waters in our capital city, basically sewage, and we have a paper that is probably out in a couple of weeks. It's the preprint available if anyone wants to read it, in which we analyze across 2 years how the microbiome from the urban environment is influenced by human microbiota, and how this impacts on the dissemination of AMR. And we found very interesting patterns in the dissemination of AMR genes to carbapenem antibiotics correlated with the development of different outbreaks in hospitals in which the patients are delivering part of their microbiome through the pipes, and this of course reach the environment and have an epidemiological impact in how our closer environment is composed, and how it could, in the future, can impact in our health. So this is something that we are doing, but also the water sources or other sources of different microbiotas could be very interesting to analyze together with the human microbiome.
Yep. Perfect. Thanks.
And actually, when you look at the species composition in the gut of the most isolated and rural population and individuals, you can already spot a lot of species that are clearly environmental bugs. Actually, so we need to work on that, and we're probably missing a lot of these species that are maybe not exactly not exactly human gut microbiomes, but just environmental bugs just passing through because less sanitation, or source of water and diet, for example.
Yeah, perfect. Thanks so much for answering that. And we have a question from Sunda, which I will take the liberty to reformulate a little bit. So what are the possibilities of harnessing microbial metabolism within human hosts and alleviating, mitigating in general, maybe I will ask for more general like disease or like improve health, especially in underrepresented communities.
Is that for me? Because I'm not sure I get the question, sorry.
Oh, sorry. What are the possibilities of harnessing the microbial metabolism, or maybe the work you have done in general to maybe improve health as well in those communities?
I see.
Mm-hmm.
I think diet is a good start. Improving the source of complex fiber, for example, will be a good start. We know how it's important for many bacterial species that are missing in the gut of industrialized population, and species that potentially have high impact on human health. So yeah, I think that could be a good start.
Yep, Dr. Iralo, feel free if you want to answer that as well, or
sorry, because I was trying to answer another question.
Oh, sorry. Yeah,
in
just
the chat, but
yeah,
no problem.
I do the same. It's not easy to follow.
yeah, sorry.
That's exciting.
Yeah, the question would be also for Dr. Iralo. Do you think that some of the work you have done, for instance, on the Latin Biota project could be like leveraged to maybe, create more specialized interventions to the microbiome, specific for Latin America, for instance, so that like intervention that would work better for that population than, for instance, trying to just extrapolate from, known things that we know work for like Northern American or European populations.
Yeah, indeed, this is our longer term goal, to provide a resource and a new knowledge that can be used to improve regional or local interventions that can be adapted to what is normal for our populations. And for that reason is also that in this second phase of the project, we are starting to focus on the analysis of different diseases that are prevalent here, because, for example, we can have microbial biomarkers for certain diseases that are not the same for Latin American people that for African, European, or US populations. So yes, the answer is yes.
Yep, perfect.
I spotted a question that I didn't answer yet, if that's helpful.
Yeah, definitely. Go ahead.
From Segman, what could explain the improvement of cholesterol removal by changing from single culture to co-culture? The hypothesis, again, that's just an hypothesis that we have. So when we used just the EcoPro, so the first cholesterol converter strains, we actually very quickly, after a few days, usually 7-8 days, we reach a plateau. Even if you had more of the strain, we reach a plateau and we cannot turn more cholesterol to coprostanol. However, when we add the Ocidis spiraceae in the co-culture, then both sterols and all the sterols in the media decrease. So the hypothesis is that actually the first PC turned cholesterol to coprostanol, and the second one used the coprostanol generated in the media and reduced this to something else, not a sterol or not something that we can detect, but that helps to, continue the process and the metabolism and to reduce the overall sterol content of the media. That's the idea. We gonna test for that easily by replacing the cholesterol by coprostanol in the in vitro experiment. So hopefully we will have an answer for this question pretty soon.
Perfect. So we have a question by Sean again to both speakers, and he's asking, how have you thought about partnering ethically and equitably with underrepresented population in your research?
That's very, very important aspect, I think. And in particular, we didn't yet, but we are open to do it and also keen on doing that. And of course, if if there is some idea or how to reach this, I would be very grateful to hear, because it's something that we didn't do because we prioritize the the core activities like sampling, sequencing, analyzing data, and indeed we are now working very hard to get the first paper published from this whole data set, but in the longer term this is very important, and it can help the project itself a lot.
Yeah. So on our side, so I think there is a lot of levels where you can actually be careful. So I'm just giving an answer of explaining what we do. I don't say that's what we should all do. We're trying to do science the right way. We're trying to do the best we can, but we are also very open to any suggestion, and we're open to learning more about the best practice. But I think the first very important thing is to avoid what's called parachute science. So I think having collaborators in each country where you actually work is extremely important. You have to have local collaborators, local scientists in each country because they know the country, they know the population, and they should also be part of advancing the microbiome science from their respective countries. So that's the first step. Then what we do when working with underrepresented population, so indigenous population in some case, saying that the consent form is an important step. We strongly believe that having consent form, written consent form in the local language is extremely important to give as much information as possible to the participants, so they know exactly what's going on, why we are here, trying to collect some samples from them, etc. And that's also on the same topic, what we are trying to do, and we just started in a few countries, but we really hope to be able to do that in all the countries we work with. We also think it's important to get back to the population after you actually do the work, after you actually generate the data and you the results, and you publish the paper. You have to go back to the population. You have to go back explaining what you found and why participating to this study was so important. And I think it's just trying to be respectful to the participant and everybody. There is many aspects that could play a role here. I'm probably missing a lot, but that's already an overview.
No, that's perfect. Thanks for the replies. There were two questions for Dr. Poyet, and they're both related, basically, which mouse model was used and whether those were germ-free mice or were treated with antibiotics to level the microbiome.
So I'm certainly not saying that one model is better than the other, but so far we used regular mice after antibiotics treatments. That's what we did, but there is pros and cons in both models. You should not just listen to my answer, just see with who you are collaborating, see if using a model or another can help answering your question, but
Perfect. And I also had another question for Dr. Iralo. And in the beginning of your talk, you also alluded to that Latin America is maybe one of the continents, one of the most complex migration histories as well. So how do you think that is kind of, I mean, you already showed some migration patterns that were obviously, but do you think that's has a particular influence on the microbiome maybe compared to other continents or populations?
Well, I think it has indeed we performed some global scale analysis with the whole dataset, in which we found strong signals and strong correlation between the distance in terms of microbial diversity between individuals and their geographic distance. So that all supports and adds more on the concept that different regions in the world and different populations have their own patterns of variation, and so the migration patterns can affect that. There is some very interesting paper, I don't remember, it is published in Cell, I think, I don't remember now the authors, about how the different generations of immigrants from Southeast Asia to the US, their microbiota is altered generations after regeneration from the date they immigrated into the country. So we didn't measure for our data set because we don't have much information about the ethnical or genetic background of the participants and the donors. This is a weakness of our first data set, but for sure it is important, and there is something there, and Latin America is very complex. We have had during centuries a very important influence from slavery from Africa, also in the Pacific coast, countries like Peru have or had an immigration, a very strong immigration from Japan and from Asia. So there is very, very complex. And today, what we found probably is a picture of all this mixture across many years. And also these immigration patterns have influenced a lot today's diet in these countries, because for example in Peru or in Uruguay, our regular diets are absolutely different.
Yeah, thanks so much. So I think we have now taken almost 40 minutes in the panel session. So I think I'll give our speakers their freedom back and thank them so much for taking all the time with us, and it was really very illuminating, amazing talk. So a huge virtual applause again to our two speakers today. And yeah, I think that was a really great start for the symposium.